Hello to all, my name is Guy, and I would like to thank the organized committee for the opportunity to present our work that deals with blueberry growth in calcareous soil as a model for other species that prefer acidic environment. World blueberry production has increased in the last decade thanks to increasing demand for this healthy food. Most of the growing area of blueberry were located in North of America, but in recent years, the growth has spread to other parts of the world. The soil conditions in part of this region are non-optimal for commercial blueberry production. Blueberry is a classophog plant, therefore the optimal soil conditions for blueberry cultivation are low pH, high aeration, and high content of organic matter. This means that calcareous soil that cover about 50% of earthland area are not suitable for blueberry cultivation. Calcium carbonate has the highest capacity to buffer pH reduction in the soil. Therefore, the pH level of the calcareous soil are mostly above 7. For illustration, we can see a representative picture of blueberry plant that grew in optimal condition of pits filled by mixture of tuff plus peat compared to one that grew in calcareous clayey soil. Previous study suggested that inhibition of blueberry production under non-optimal pH condition is a result of nutrient deficiency that are the blueberry growth in acidic soil where the ammonium is the major ionic form of nitrogen. Therefore, plants prefer ammonium than nitrite. And nitrite is the major form of nitrogen in alkaline soil. And the second is micronutrient deficiency, mainly of iron. However, to best of our knowledge, the mechanism which lead to inhibition of biomass production of blueberry in neutral to slight alkaline pH soil is still unknown. The main objective of this study were to evaluate the combined effect of different proportion of ammonium among the total applied nitrogen and soil calcium carbonate content on biomass production and mineral uptake of blueberry. And the second one was to evaluate the combined effect of pH and calcium concentration on root morphology and function. In order to evaluate the combined effect of ammonium proportion and calcium carbonate concentration on biomass production and mineral uptake of blueberry plants were growing in 10-liter pots filled by sandy soil. The treatment were consist 4 levels of calcium carbonate, 1%, 5%, 10%, and control with no addition of calcium carbonate, combined with 3 different proportions of ammonium, 33, 66, and 100%. The concentration of all of the necessary nutrients were similar in all treatment and were applied by a fertigation system. Nutrient concentration in diagnostic leaves were measured several times along the growth season. And that in the canes, at the end of the growth season, biomass production was measured at the end of the growth season. The pH of the leachate were measured several times along the growth season. In order to evaluate the combined effect of the pH and calcium concentration on root morphology and function, blueberry plants were grown in nutrient solution at 2 levels of pH, 4.5 or 7.5, combined with 5 concentration of calcium. The concentration of all of the rest necessary nutrients were similar in all treatment. Biomass accumulation, chlorophyll, and mineral concentration were measured after 3 months. Nutrient accumulation and their uptake per root unit were calculated. Microscopic observation and analysis of root surface were done too. In the next slide, I will present you a combined effect of ammonium and calcium carbonate treatment on the pH and the plant parameter. This figure shows the changes over time in x-axis of the pH and y-axis of the leachate in 33% of ammonium in 4 different concentrations of calcium carbonate. The color of the symbol in the legend are representing the calcium carbonate concentration and will be similar in all the following figures, where the blue color represents control treatment, green 1%, red 5%, and orange 10% of calcium carbonate on weight basis, at 66% of ammonium and at 100% of ammonium. In general, it could be seen in 33% of ammonium, the pH of the leachate decreased to the level that are suitable for growing blueberry, which representing by purple line, only in calcium carbonate free soil treatment, after about 90 days from start of application. At the 66% of ammonium, the pH of the leachate decreased to the level that are suitable for growing blueberry even in soil that contain 1% of calcium carbonate, at the same period of time. At 100% of ammonium, the decrease of the pH is much faster than that presenting in 33 and 66% of ammonium in these two concentrations of calcium carbonate. In addition, after about 1 year from start the application, even in soil containing 5% of calcium carbonate, a level of pH that was obtained is suitable for growing blueberry. However, in soil that contains 10% of calcium carbonate, it's impossible to affect the pH of the leachate even at maximum proportion of ammonium. The effect of ammonium proportion and calcium carbonate concentration treatment on the biomass production of shoot is presenting in this table, which shows that an increase in proportion of ammonium from 33 to 66% was associated with increase of biomass production, and further increase of the proportion to 100% was associated with slow increase in biomass production. Overall, the increase of the proportion of ammonium from 33 to 100% lead to significant increase of biomass production. An increase in concentration of calcium carbonate in the soil from 1% to 5% was associated with significant reduction in biomass production. Another way to present the combined effect of treatment on biomass production is presented in this figure, which shows the relationship between the average pH of a leachate in x-axis and the relative biomass production in y-axis. The accumulation of biomass in the shoot decreased linearly as the pH of the leachate rises above 5.5, where the color is representing the concentration of calcium carbonate treatment as shown earlier. The shape of the symbol in the legend are representing the ammonium treatment and will be similar in all the following figures, where the circle representing 33% of ammonium, square 66%, and triangle 100% of ammonium. In all the calcium carbonate treatment except in soil without calcium carbonate, as the proportion of ammonium increases, the production of shoot biomass increases. In calcium carbonate free soil treatment, the average pH were lower than 5.5 in all the ammonium proportion treatment. Therefore, there is no effect of ammonium treatment on relative biomass production. This relation between the pH of a leachate and biomass production was also shown in the relation between the pH of a leachate and relative water uptake in different experiments which done by us employing another blueberry cultivar which grown in different growth medium under acidification by elemental sulfur or sulfuric acid or ammonium proportion presenting by purple rhombus symbol. In general, the concentration of manganese in the leaves in y-axis increases with the decrease of the pH in x-axis. But in any given concentration of calcium carbonate, which represented by specific color, as the proportion of ammonium increases, the concentration of manganese in the leaves increases. The clearest effect of the ammonium treatment on concentration of manganese in the leaves was expressed in the higher calcium carbonate treatment, which represented by orange and red color, where the manganese concentration were increased without a significant change of the pH, a phenomenon which indicates that the acidification process in the rhizosphere is more efficient than that of the bulk of the soil in terms of manganese availability. The increase of manganese concentration in the leaves in x-axis was related to increase of biomass production in y-axis. The concentration of calcium in the canes in y-axis increases with the increase of the pH in x-axis. The highest concentration of calcium in the canes were presenting in the highest calcium carbonate treatment. The increase in calcium concentration in the canes were relating to decrease in relative biomass production. To summarize the presentation till now, as we have shown in the last slides, low manganese concentration in the leaves and high concentration of calcium in the canes were associated with decrease of biomass production. In the next slide, I will be presenting the combined effect of the pH and calcium concentration on root morphology and function. The effect of a pH treatment on growth parameter was significantly greater than that of the calcium concentration. Hence, only the effect of the pH treatment will be presenting. The biomass production of each of the plant organ and that of the whole plant were significantly higher in 4.5 and 7.5. as well as chlorophyll concentration in the leaves. The concentration of manganese in the shoot was higher in acid pH, but in the root, the concentration of manganese was higher in alkaline pH. Indicates that manganese translocation from the root to the shoot was inhibited under higher pH. The concentration of iron in all the plant organs was higher in alkaline pH, opposite to effect of the pH on chlorophyll concentration, which indicates that inhibition of the reduction reaction of iron in the leaves that probably occurred. In this slide, I will present the microscopic analysis and observation. The concentration of calcium and phosphorus were higher in root of plant growing in alkaline pH. The root surface in acid pH visually look clear in 3 levels of calcium concentration, 0, 10, and 100 milligram per liter. But in alkaline pH, the root surface were not visually look clear. The calcium and phosphorus were accumulated on root surface at alkaline pH. This accumulation was increased as the concentration of calcium in the solution was increased. The tip of the root in acid pH visually look clear, but in alkaline pH, the tip of the root look damaged. The higher concentration of calcium worsens this damage. The effect of the pH on mineral uptake per root unit does not have a consistent trend, which may indicate that the pH does not have negative effect on element uptake by the roots. Our results indicate that 2 major mechanisms could be associated with the inhibition of plant biomass production at alkaline pH. One of them is direct morphological damage of root tips, which inhibited the root elongation. This effect was combined by accumulation of calcium and phosphorus on root surface, which became more significant in higher concentration of calcium. Second one is manganese and iron deficiency. One of them is inhibition of manganese translocation from root to the shoot, and the second one is inhibition of reduction reaction of iron in the leaves. Overall, the concentration of calcium had no significant effect on biomass production and on the other measured parameter of blueberry plant. The result presented in the solution experiment in terms of the negative effect of the pH on the manganese and chlorophyll concentration and biomass production are similar to those obtained in the pot experiment. High proportion of ammonium among the total applied nitrogen and enhanced biomass production of blueberry in soil containing low concentration of calcium carbonate, probably due to direct acidification of rhizosome. Thanks to your attention.